And now, an exclusive sneak preview of a recent X-Talks webinar, Immunotherapy for Pancreatic Cancer, Peptidic Targeting of a Tumor-Specific Glycan.
So one of the ways that we're also utilizing MGS5 is to deliver targeted antigen-loaded liposomes, or TAL. So when we look at the current immunotherapy landscape, we have a distinct difference between responders and non-responders of immunotherapies, with responders generally being classified with these hot tumors, or tumors that have high microsatellite instability leading to increased neoantigens, as well as they have a tumor microenvironment that's very favorable to immune infiltrates. And these generally respond, again, really well to immunotherapies. However, when we look at like pancreatic cancer, they're generally classified as cold tumors. And so they lack microsatellite instability. So they're not having these high levels of neoantigens being produced, as well as they have highly immunosuppressive tumor microenvironments. So these immunotherapies have really struggled to show any therapeutic efficacy within these types of tumors. And so there's an urgent need to develop a novel immunotherapy to target current non-responder patients by converting non-inflamed and immune-excluded tumors into treatable targets. So what we've done is, again, we are utilizing our targeted antigen-loaded liposomes, and TAL has three modular components. We have our tumor-targeting MGS, which in this case we're utilizing our MGS5. It has a pegylated stealth phospholipid layer, as well as our synthetic H250 peptide that's encapsulated within the liposome. And we've chosen H250 because it's a sequence derived from the measles hemagglutinin protein and is known to generate a strong CD8 positive T cell response. It's also HLA-A2, has a strong affinity for HLA-A2, as well as shown affinity for the H2 MHC class 1 haplotypes to allow us to detect the efficacy of TAL within murine mouse models. But what we're really trying to do here is to exploit the recall immunity by tricking the immune system to recognizing tumors as a pathogen-infected cell. So again, our mode of action is we're utilizing chemically synthesized H250 peptides. We are then encapsulating and loading it into our liposomes, which again have our MGS5 on the outside to be able to then home and target and internalize into the tumor cells. That then allows for the release of the H250 peptide within the tumor cell to then be presented onto MHC class 1 and to present to T cells. This triggering the patient's immune system to clear the tumor. So much like what we've done with MGS5 in the past, is we're able to visualize via confocal microscopy and flow cytometry the internalization of actually the H250 peptide. So rather than having MGS5 be conjugated to 647, this time we are conjugating our H250 to 647, and that's depicted in red. Again, our green is our cell membrane stain, and then the blue is the nuclear stain. And so when I refer to NTP, NTP is our non-targeting peptide. So these are liposomes up in the left-hand corner where they actually are lacking the MGS5 on the outside. Again, so they don't have that targeting capability. And so what we see is we see no uptake of our H250 647. However, when we look at the TAL-treated cells, what we see is our H250 647 internalized into the cells. And so again, this tells us that MGS5 is allowing for the internalization of the liposome and the release of the H250 peptide. Again, we're able to quantify the number of H250 molecules internalized per cell. When we look at the quantification of our NTP, again, these are the liposomes that lack the MGS5 on the outside, we see very low numbers of the H250 molecules internalized per cell. However, with increasing amounts of our liposomes that have the MGS5 on the outside, what we see is a significant number of H250 molecules internalized per cell, upwards of 3 million molecules of H250. And so again, this really tells us that MGS5 is necessary for the internalization of TAL. Additionally, we wanted to look at whether or not MGS5 is able to deliver TAL, leading to H250 presentation on MHC class 1. So we utilized an interferon gamma secretion assay. On the left, I've highlighted that we're using a human cancer non-small cell lung cancer line that is HLA-A2 positive, and then utilizing PBMCs from measles vaccinated patients. And so we've taken our PBMCs and co-cultured them with our cancer cell line, and then added either a blank, which is our MGS5 targeted liposome, but it has no H250 on the inside, or incubated with NTP, which again is the liposomes that contain the H250 but don't have the MGS5 on the outside, or with TAL, where again we have the whole complex. We have MGS5 targeted liposomes and then loaded with the H250 antigen. And what we see with the blank and NTP is very low levels of interferon gamma secretion, but with the addition of TAL, we see a significant increase in interferon gamma secretion. We've utilized H250 free peptide as a positive control to show that our H250 is able to induce interferon gamma secretion alone. And then when we deplete the CD8 positive T cells, we see a complete abrogation of interferon gamma secretion. And this really tells us that the CD8 positive T cells are the ones mediating the interferon gamma secretion. Again, we've done the same experiment, but this time in a murine cancer cell line that's H2KB positive. We've taken lymphocytes from a vaccinated mouse, and again, utilizing blank liposomes, NTP liposomes, and then our full TAL, we see a significant increase in interferon gamma secretion with the TAL treatment compared to the blank and NTP. Again, our H250 is our positive control to show that H250 is able to induce an interferon gamma secretion. And then again, when we deplete the CD8 positive T cells, we again see that abrogation of interferon gamma secretion. And so this really tells us that MGS5 is able to deliver TAL to targeted cancer cells and allows H250 to be presented on to MHC class 1.
To view the entirety of this webinar, visit the link in the description and register on xtalks.com completely free of charge.